DNA extraction process. DNA extraction is the process of isolating DNA from cells or tissues. This procedure is essential for many fields of research, including genetics, forensics, and biotechnology. The DNA extracted from cells can be used for various applications, such as PCR amplification, sequencing, and cloning. The DNA extraction procedure involves several steps, including cell lysis, DNA purification, and DNA quantification. The first step is to break open the cells to release the DNA. This is achieved by using a lysis buffer containing enzymes and detergents. The enzymes break down the cell wall, and the detergents disrupt the cell membrane, releasing the DNA into the solution. Once the DNA is released from the cells, it must be purified to remove contaminants such as proteins, lipids, and RNA. This can be done by using various methods such as phenolchloroform extraction, silica-based column purification, or magnetic bead-based purification. Phenolchloroform extraction is a widely used method for DNA purification. In this method, the DNA is extracted from the solution using an organic solvent such as phenol and chloroform. The organic solvent separates the DNA from the other components in the solution, which can then be removed by centrifugation. Silica-based column purification is another common method for DNA purification. In this method, the DNA is bound to silica beads, and contaminants are washed away using various buffers. The purified DNA is then eluted from the column using a low-salt buffer. Magnetic bead-based purification is a newer method that uses magnetic beads coated with a DNA binding agent. The DNA binds to the magnetic beads, and contaminants are washed away using various buffers. The purified DNA is then eluted from the magnetic beads using a low-salt buffer. Once the DNA is purified, it must be quantified to determine its concentration and purity. This can be done using various methods such as UV spectrophotometry, fluorometry, or gel electrophoresis. UV spectrophotometry is a widely used method for DNA quantification. In this method, the DNA absorbs light at a wavelength of 260 nanometers, which can be measured using a UV spectrophotometer. The absorbance at 260 nanometers is proportional to the concentration of DNA in the solution. Fluorometry is another method for DNA quantification. In this method, a fluorescent dye is added to the DNA, which binds to the DNA and emits fluorescence. The fluorescence is measured using a fluorometer, and the intensity is proportional to the concentration of DNA in the solution. Gel electrophoresis is a method for DNA quantification and purification. In this method, the DNA is separated based on size using an agarose gel. The DNA is stained with a dye such as ethidium bromide, which fluoresces under UV light. The DNA bands can be visualized using UV light, and the intensity of the bands is proportional to the concentration of DNA in the sample. In conclusion, DNA extraction is a crucial procedure in various fields of research. The procedure involves several steps, including cell lysis, DNA purification, and DNA quantification. The choice of method for each step depends on the sample type, the downstream application, and the resources available. The quality and quantity of DNA extracted are essential for the success of downstream applications, and thus, care must be taken to optimize each step of the DNA extraction procedure. Thanks for watching this video. Remember to like and share our videos, and leave your comments below. We'd love to hear from you.